All right, this is chapter 5, the integumentary system for anatomy, Biol-16. So the integument is the largest organ of the body. The integument is basically your skin, and it's very, very important. It keeps us from dehydrating. It protects us from the environment. It keeps us warm, protects us from bacteria, and has a lot to do with our appearance, which is a part of human communication. So let's talk about the integument. The integument can be divided into a couple different layers. The cutaneous membrane, which is the skin, the skin that we're familiar with on the outside of our body. The skin itself has accessory structures, which would consist of the hair, nails, and glands that produce sweat and oil, and also the mammary glands. We're going to talk about the mammary glands in the chapter on the reproductive system. Now, the cutaneous membrane, the skin, is made up of the epidermis and the dermis. The epidermis is superficial, or on top, and the dermis is deeper underneath the epidermis. So in terms of what we're going to be looking at, we're going to look at the epidermis and dermis separately, and the subcutaneous layer. The subcutaneous layer is underneath the epidermis and the dermis. It's a layer of connective tissue that allows the epidermis and dermis to hold on to the underlying tissue, like muscle and so forth. And it's responsible for pressure sensation, deep pressure, like when you have a rubber band wrapped around your wrist. Again, more important functions of the integument is protection from physical, chemical, and pathological threats, detecting conditions of the environment like temperature and pressure, temperature regulation. So when we get hot, we sweat. There's also an increase of blood flow to the surface, which allows us to cool off. Vitamin D synthesis. Our skin makes vitamin D. Oh, excuse me, my goodness. My apologies. And vitamin D is essential for absorbing calcium into our systems. And the integument gives us information about the health, the age, and the emotional state of others, in that our health is reflected in our skin by whether or not your skin is dry or cracked or smooth and so forth. So this picture here, figure 5.2, is a very, very important picture. This is a very simple drawing of the layers of the skin. It's going to get more detailed, but you're going to be seeing something like this many times in this course. The reason being is because all of the 4 major tissue types are found inside of the integument. So we have the epithelial tissue, which is the skin on top. We have muscle tissue associated with the erector pili muscles. Here's a piece of smooth muscle that allows our hairs to stand up when we get scared or when we get cold. So that's epidermal, or epithelial, sorry, muscle tissue. We have connective tissue down here. You're seeing adipose here, but there's also reticular connective tissue, and nervous tissue. Right here we have a positium corpuscle, which is a detector for deep pressure. And then we also have free nerve endings here. Free nerve endings are associated with pain and general sensation of the skin. So this model is going to be very, very important. You're going to be seeing this again and again and again. Now, in terms of the layers of the skin, the simplest way of breaking it down is the epidermis, the dermis, and the hypodermis. This would be superficial, deep, and deepest, all right? The epidermis is made up of, on top, the squamous epithelial layer, the skin cells that slough off. It goes down to a cuboidal layer that we'll talk about later. And then you hit the dermis. The dermis consists of connective tissue, areolar, loose connective tissue here, close to the epidermis. And then you're going to have lots of reticular tissue down here, lots of collagen fibers that help strengthen the skin. Deeper down in the hypodermis, again, is where you're going to find lots of other connective tissue like adipose tissue, but you're also going to find the main blood vessels, the cutaneous vascular plexus, all right? Good thing to know. The cutaneous vascular plexus brings blood in to nourish hairs, also to nourish the tissues lying above, and the veins drain blood out. We'll talk a little bit about the circulation to each individual section of the skin in a little bit. So let's start with the cutaneous membrane, the epidermal layer. The epidermal layer consists of 4 different cell types: keratinocytes, melanocytes, Merkel cells, Langerhans cells, that are all distributed at different depths throughout the cutaneous layer. So here's the surface. The cutaneous layer consists of the striatum corneum. Okay, so if we come back here, it's up here in the epidermis. Striatum corneum has lots of keratinocytes. That's what these flat cells are. They can be either kind of round while they're still alive or flat and squamous as they die. We have the striatum lucidum. The striatum lucidum is going to contain lots of melanocytes. The striatum granulosum underlies the striatum lucidum. The striatum spinosum. The spinosum is basically the freshest of the new cells coming up through the epidermal layer. And then we have the striatum germinativum. This is where the stem cells that make new skin reside. The striatum lucidum contains lots and lots and lots of melanocytes that make melanin. Now, you might find a few melanocytes a little deeper. Don't worry about that right now, okay? That's why you see this dark line here. Underneath the striatum germinativum, you're going to find the basal lamina. The basal lamina is basically kind of a gooey matrix that the striatum germinativum sticks to using hemidesmosomes. Remember those tight joinings? Underneath that is the dermis. So here in the dermis, you're going to start seeing some parts of Merkel cells. You'll see parts of Merkel cells over here as well, and maybe some Langerhans cells, which we'll talk about in a few minutes. So from deep to superficial, now we're going backwards. Striatum germinativum, striatum spinosum, striatum granulosum, striatum lucidum, and striatum corneum. We're going to talk about what makes each one of these special. Now, from top to bottom, corneum, lucidum, granulosum, spinosum, basal, or striatum basalis is also striatum granulosum, just so you know. Here's a melanocyte. Here's the dermis, here's a Merkel cell, and a sensory neuron. So this is just another drawing from your book to identify the depths at which you're going to be seeing each one of these cell types, what they look like. One of the things I want to point out to you is that the desmosomes are going to be found down here in the striatum spinosum, which makes them look kind of spiny, okay, because they have lots of desmosomes adjoining each cell to its partner next to it. Striatum germinativum, this is attached to the basal lamina. Like I mentioned, lots and lots of stem cells that kick off the keratinocytes, skin cells. Melanocytes here, what they do is they synthesize melanin and then they inject it into the keratinocytes as they're wandering by. Isn't that nice and convenient? Merkel cells themselves are pressure receptors that are sensitive to touch. They do release chemicals like histamine to stimulate other adjacent sensory nerve endings to tell them, hey, something bad has happened here. Now, something that I forgot to tell you here that I'm glad I wrote it down here. In some individuals that are affected by a genetic condition called albinism, these individuals have lower melanocyte activity. What that means is that they have the same number of melanocytes, but the melanocytes aren't producing adequate melanin to demonstrate pigmentation of tissues. So there are different degrees of this in albinism, but some people with albinism don't produce hardly any melanocytes at all, or melanin. The melanocytes they don't produce melanin because of a genetic mutation, and that results in their skin looking very pale. Usually their hair is very blonde, and sometimes the iris of their eyes are kind of reddish. Albinism occurs in many different vertebrate species. If you just think about white rabbits with red eyes, those are albino rabbits. I just wanted to show you a slightly more detailed photograph, or we'll call this a histological preparation, because remember, the study of the body tissues using stains is called histology. So this histological micrograph here is showing us melanin pigment inside of melanocytes in the striatum germinativum. This might be a stem cell right here. Some of the stem cells, here's the basal lamina, that might be a stem cell. Anyways, these melanocytes, what they're doing is they're producing melanin and kicking it off into individual keratinocytes. Inside of the keratinocytes, the melanin sometimes gets localized to these small granules called melanosomes. So these melanocytes branch out and they kind of have fingers all throughout the tissue. All right, we're going to move to the striatum spinosum now. The striatum spinosum now is overlying the striatum granulosum. And so when a stem cell divides, the daughter cells get pushed up and become keratinocytes. Now I'm going to pause for a minute here and draw something out here, if my pen will work. So let's pretend that this is a stem cell. When the stem cell divides and gives rise to two new cells, one of the two cells is going to go off to differentiate as, in this case, keratinocyte. The other cell regenerates itself as a stem cell. So that's a part of the definition of what is a stem cell. When the stem cell divides, it produces a new offspring, a new daughter cell that differentiates into something, but then it reproduces itself. So stem cells are going to kick off a bunch of keratinocytes. Let me erase this here. And those keratinocytes start migrating upwards through the other layers, and eventually they produce these protein bundles called tonofibrils to form desmosomes. I'm sorry, I should fix that. Those desmosomes are going to connect one keratinocyte to another so that all the cells have a tight network holding them together. This is a part of our waterproofing, a part of our protection from the environment. If you look at these cells in a scanning electron micrograph, those tonofibrils look real spiny, hence striatum spinosum, not spumoni. Oh my gosh, some of you will get that. Melanocytes and Langerhans cells. Langerhans cells are a part of the immune system. These things are phagocytes. They go around and they consume dead cellular debris, any bacteria or viruses that they run into. So melanocytes and the Langerhans cells are what you find within the striatum spinosum, kind of in the lower layer. Some cells continue to divide in this layer, but for the most part, that's all happening over in the a little deeper in the striatum granulosum, which is right here. Okay. So after the striatum granulosum, as you go deeper, there's no more mitosis happening. No more cell division really happening unless there is an injury. And then the underlying tissues will replace and form scar tissue. So keratinocytes become displaced from the striatum spinosum. Man, I guess I want some ice cream. Spumoni, geez. Making our keratin. And there's another protein called keratohyalin. Keratohyalin, do not confuse this with hyaline cartilage. It is not cartilage. These are granules that kind of harden around the keratin filaments inside of the keratinocytes, and they show up as these dark kind of grainy things when you look at an electron micrograph. As the cells start migrating upwards, they become flatter, more squamous. The membranes thicken, organelles in the nucleus disintegrate, and the cell basically dies, leaving just the keratin fibers and the granules, the keratohyalin granules, sitting there in a membrane, basically representing an old membrane. And it looks kind of like, I always kind of imagine it kind of like a chain link fence, one chain link fence stacked on the other. Keratin can also become nails and hair in humans. If you're thinking about animals, it can become a cow horn, whale baleen, snake scales, bird feathers, and also bird beaks. So keratin is a really strong protein. Now, moving to the striatum lucidum. This is in thick skin. We're going to be defining thick versus thin skin now. This is the skin that covers the granulosum. Okay, it's flat, really densely packed cells, full of keratin. These cells don't stain very well in slides, hence that's why it's lucidum. Basically, you have 15 to 30 layers of flattened interlocking cells along a surface in the corneum, and then underneath that you would have the lucidum. So let's look at a picture here. We want to look at thick skin versus thin skin. On the right, we have thick skin. On the left, we have thin skin. Up here on top, this is what you're looking at in terms of thin skin. There's not a whole lot of tissue. Thin skin is what you find over most of the body surface. On the other hand, on the palms of your hands and on the soles of your feet, you'll find much thicker skin. In other words, the striatum lucidum is much thicker. So let's come over here and look at thick and thin skin in terms of the connections between the epidermis and the dermis. So remember, the epidermis is the top layer. It consists of all the striatum. Here's the striatum corneum on top, the striatum lucidum here. It's kind of, this is where you'll see like those, this is going to be thicker in the thick skin. Here's the dermal layer down here. The way the dermal layer connects to the epidermis, okay, is through these little zigzags called dermal papilla. You can think about it as kind of like Velcro. these epidermal ridges intercalate into the dermal papilla and they stick together, holding the epidermis onto the dermis. When you're looking at thick skin here, you'll see a real thick basal lamina. The striatum lucidum is barely noticeable, but over here in thick skin, the striatum lucidum is very, very obvious. So the big difference here between these two, look at the striatum corneum, huge here, very small here. So the lucidum is the defining feature for whether or not the skin is thick or the skin is thin. What might you see on an exam, thin versus thick? I would probably ask you about the striatum lucidum here, okay? Oops, sorry, let's go back to this former picture. What's the difference? Why do you think it's different between thick and thin skin? And another thing to bear in mind is, why on earth would you have a whole lot of thick skin on areas where there's a lot of rubbing? Say, for example, in a callus, okay? I'm going to let you ponder that on your own. Okay, so back to the epidermis. The epidermis, I'm sorry that, I tried to blow these pictures up from your book. So please refer to figure 5.4 so you can look at the layering between all of these squamous epithelial cells that you'll see here and the deeper layers, which is what you're seeing here, the striatum corneum, striatum lucidum, and so forth. Here's another picture showing us the surface, the striatum corneum, striatum lucidum, striatum granulosum, spinosum, and the germinativum. So over here, your striatum lucidum, right? Remember, you're going to find a few, you can find a few of the melanocytes there. Do you think this is thin or thick? Think about it. All right. Cells at different layers of the epidermis are going to look slightly different depending on how old they are. Again, I would recommend that you look at figure 5.6 in your book. You will be able to see some of the differences in the cell morphology a little bit better. I tried to blow it up and it looks crummy here. My apologies. Okay, so that is the epidermal layer. Let's talk about the dermal layer. The dermis is deep to the epidermis, and it's broken down into two layers, the papillary layer and the reticular layer. I think of the reticular layer as lots of ropes, okay? In other words, lots and lots of strings of collagen and some elastin. The papillary layer is more airy. In other words, it does have connective tissue here. Okay, so there's connective tissue here, lots of ropes, which is also connective tissue. But the papillary layer has lots of capillaries, lots of papillae. It looks a little less organized than the reticular layer, okay? The Meissner's corpuscles are going to be found in the papillary layer, and remember, those are associated with pressure, okay? Shallow pressure. I'm going to tell you that right now. The reticular layer is dense, irregular connective tissue. You'll see that in a minute. This is going to surround hair follicles, glands, blood vessels, holding things in place, keeping things strong, and basically not moving around. You will find some collagen, lots of collagen here actually. Some collagen in papillary, but lots in the reticular layer. So let's look at our dermal layer here. Here's the reticular layer, and the way that this figure is drawn, I really like this because it's showing you lots of lines, lots of ropes, lots of collagen bands holding this layer together. The papillary layer right here, which is associated with the dermal papilla here are these little dermal papilla. Here's a free nerve ending and so forth. This looks a little less organized. So here's the papillary layer of the dermis. You can see that there is some collagen here, all right? You do have some tubes, you do have some ropes, but you also just have some, Merkel cells. That's not a Merkel cell. You'll see some white blood cells in here, things like that, immunity basically. And down here in the reticular layer of the dermis, lots and lots and lots of collagen. Look at that. Isn't that incredible? Lots of elastin too to make it stretchy. This is also where you're going to start connecting down to the cutaneous layer where you're going to start finding fat, okay? So the reticular layer connects to the cutaneous layer, and basically what happens is a lot of these reticular fibers intercalate into the adipose tissue holding things in place. So here's just another histological preparation showing you a papillary layer right here and the reticular layer right here. You can see the dermal papilla here, right? And the epithelial what, guys? Can you go back and look at that? What are these little bumps going down called? So let's talk a little bit about blood flow. I mentioned this in that first slide where you saw a picture of the skin. Blood vessels, BVs, form a network in the dermis called the cutaneous plexus. The cutaneous plexus is going to supply the tissues with nutrients, oxygen, remove CO2 from the area, remove waste products. And so basically it's nourishing tissues of the epidermis and the dermis and the hypodermis for that matter, okay? In the papillary layer, you're gonna find lots of capillaries. These are called the papillary plexus, okay? This is where you'll bleed a little bit if you get kind of a superficial scratch, all right? These papillary plexi is plural for plexus, are very, very important for temperature regulation. When we get really hot, we tend to flush, all right? Your face will turn red. What's happening there is these capillaries are opened up to a large amount of circulation, and that capillary circulation is going to allow your body to release heat by blood flowing close to the surface of your skin. If you get really cold, the papillary plexus aren't completely shut down, but you're not going to feel a whole lot of blood. Oh, I'm yawning again. My apologies. Circulating to the papillary plexus. You guys, I'm going to be honest. This is the third time I've recorded this particular lecture. I've had some technical issues today, and it's been a long lecture day for me. So if you hung through long enough, this was the challenge with chapter 5. I had all kinds of problems with my dumb microphone. Okay, enough of those sad stories. Nerve supply. There's a lot of nervous system innervation to the skin so that we can sense the environment around us. It controls blood flow and it helps monitor our sensory receptors in terms of pressure. Here is, I don't know why I have another picture of pigmentation this deep, but I just wanted to, I should have put this further forward in the lecture, but this is how melanocytes produce melanin and how the melanin gets into individual keratinocytes. Each melanocyte produces melanin. It buds into individual cells. These are called melanosomes. And the melanosomes, depending on how much melanin and melanosomes each cell absorbs, determines how dark the cell is. Over here on the right, you have a situation where this particular cell is not producing a whole lot of melanin. And in fact, in a light-skinned individual, the melanosomes are still there. They're just not full of melanin. So that leads to what we see on the surface of the skin, right? So moles are considered an accumulation of melanin. They can be benign and just be little brown dots on the surface of our skin, or they can be melanomas. This would be a melanoma. A melanoma happens when melanocytes start proliferating out of control and produces a small tumor, basically. Those tumors are obvious because the borders of the mole are not round. They're kind of uneven. They look kind of blue tinted underneath because the melanocytes are underneath the skin as well as on top. And they have an irregular appearance. So this particular melanoma would probably be cancerous, but this is just a little melanocyte that a doctor might watch just to make sure you're okay. Now, the opposite of looking at moles would be vitiligo. Vitiligo is a condition where sometimes this condition is the result of the immune system attacking melanocytes. If that happens, then you end up with regions of the body that experience depigmentation. So you see a lighter color of skin compared to darker skin adjacent to it. This can also happen in hair, and what that will result in are hair patches that are white because those particular hairs aren't able to produce any pigment. And most of our hair pigment can be melanin and keratin. So The accessory structures of the epidermis consist of hair, nails, and if you are a bird, feathers or a beak, and scales in reptiles. All of these structures come developmentally from the same tissues. So the hair is a structure that's basically a bunch of keratin. The hair develops down here in the hair bulb. The dermal papilla extend, remember the vascularization, extend up into the inner root sheath, hair root sheath. We have an outer hair root sheath, and then we have the hair shaft itself. Each individual hair, not all of them, but many of them are associated with what's called an erector pili muscle. When we are scared or if we get cold, these muscles contract by way of the autonomic nervous system, and that pulls the hairs upright. A sebaceous gland is usually associated with each individual hair shaft, and that makes oil or sebum, mostly oil, to keep the hair soft and pliable. And then the hair shaft itself has a medulla, cortex, and cuticle. I'll show you a picture of what those look like in just a minute. So here's a micrograph, a histological micrograph of what a hair looks like. So here's the bulb down here. Here's the shaft. Here are some sebaceous glands. And so if we look at the structure of an individual hair, there are two main structures, the root and the shaft, right? The root is down deep and the shaft is higher, okay? It's the actual hair, okay? It's the surface, it goes to the surface and extends beyond the skin. We talked about the erector pili muscle already. Now, if you look at the cross section through a hair, you'll see connective tissue, which is the sheath. You'll see this thing called the glassy membrane. The glassy membrane is just a subdivision between the external root sheath and the connective tissue. Then you have the internal root sheath, which you saw in one of the last figures. You have the cortex of the hair, the cuticle, the cuticle is here, the cortex is here, and then the medulla is in the middle. Typically, when you hear the term medulla, it's always in the middle of a collection of cells. So if you look at a cross section through hair follicles, you can see the hair shaft. Usually it'll look brownish or reddish because of the presence of keratin. The hair root, the external root sheath, the connective tissue on the outside, the internal root sheath right there, the glassy membrane, which is right there, the cortex, the medulla way deep down inside, and then something called the matrix. The matrix is going to be associated with the hair papilla, which is the circulatory capillaries. And then you have subcutaneous adipose tissue associated with and adjacent to each individual hair follicle. Nails are a lot less complicated. If you're just looking at your nail, you have the free edge of the nail, which is the part that grows out long. You have the nail itself, the lateral fold where the nail meets the skin, the lunella, the white part of the nail, the epikinin, and then the proximal nail fold. That's the cuticle, basically. And so the proximal nail fold is dips down underneath the proximal nail fold is not the cuticle. The proximal nail fold is the actual nail proper. Sorry about that. And then you have the nail root. So basically the nail grows out along the edge of the finger, the nail body, and then we clip the nail past the edge of the finger. The hypokinin is the joining of the skin next to the skin to the underside of the nail. The phalanx is the bone. And then you have dermis and epidermis associated with tissue underlying the nail. Creepy idea, cut the nail in half. You'll see the phalanx, you'll see underlying tissue, and then you can see that the nail itself, the nail body, is supported by the nail bed that helps support the nail itself. What would you need to know for the exam? Just the basic anatomy of the organization of a nail. Same with the hair. Now, the last thing we're going to be talking about are glands. So we'll start with the sebaceous gland. Sebaceous glands are holocrine glands. I'll let you look at that type of secretion from a former chapter on your own. Holocrine glands, in this case, secrete sebum or oil. It's important for lubricating skin, keeping keratin, I guess I would say kind of not moist, but structured appropriately. And it also has antibacterial properties. So sebum is an important way, one of the important ways that our body actually fights bacterial infection. Each one of these glands is connected to a hair follicle, and the sebum moves up throughout the hair shaft. Now, sebaceous follicles, which we're seeing here, that's a gland. These are follicles over here. Secrete oil, sebum, onto the surface of the skin for all the same purposes that we just talked about with the sebaceous gland, for antibacterial properties, lubricating the skin, and so forth. Now, an eccrine gland is a sweat gland. An eccrine sweat gland releases sweat, which is mostly water, onto the surface of the skin to allow us to undergo evaporative cooling. This type of sweat is mostly involved in temperature regulation. There are other types of sweat glands called apocrine and merocrine sweat glands that are important for other jobs in the body. So myoepithelial cells contract, squeezing out the secretions onto the surface of whatever, okay? Here, the cross-section of an apocrine sweat gland, my apologies, is producing salty sweat. But a merocrine sweat gland does something different. Merocrine sweat glands produce a different type of solution. We'll talk about that in just a second. So if we talk about our apocrines here, released into hair follicles by a merocrine secretion method. You look that up in the last chapter. The apocrine sweat gland you're going to find in the axilla and the areola and in the groin regions of all humans. These are producing a different type of sweat, not the salty, watery sweat, but this is a viscous, cloudy, odorous sweat. This helps support our friendly bacteria that live in these regions and helps kind of keep yeast from growing there. Sometimes apocrine sweat glands contain pheromones, depending on where you're looking and what point in the person's life. The merocrine sweat glands, again, these are the types of sweat glands that are going to produce salty sweat. You find these on the palms of the hands, the soles of the feet. These are a little bit different than just regular thermoregulation sweat glands. These are these respond to autonomic stress events. If you think about when your hands get sweaty, those are all merocrine sweat glands doing that. They don't penetrate very far into the dermis, and basically you're just sweating out lots of salts when you get stressed. Now, there are some other modified sweat glands in the body, which consist of the ceruminous glands in the ear and the mammary glands. Mammary glands are apocrine glands. We're going to talk about those in the reproductive system. So you don't really have to worry about that for this lecture, for this chapter or unit. The ceruminous glands, the only thing you're going to really hear about them is that they have a large, they're modified merocrine glands. They have a big lumen, and including water, they secrete pigments and lipids into their compounds that they secrete. What that does is it produces kind of a waxy concoction that traps particles in the ear auditory canal and allows the removal of those particles so that you don't have a blockage. Now, sometimes in some people, depending on the shape of their auditory canal, they can end up with a buildup of auditory cerumen. Cerumen is the science word for earwax. And so sometimes people have to have that removed because the cerumen can build up and actually block auditory signals to the brain. And in general, skin, as we age, changes pretty significantly. It starts to display noticeable signs of aging early, like even in the 20s, you'll see some slight drying and so forth. It loses elasticity and thickness over time, so that by the time we're older, our skin is thinner, it may wrinkle more easily, there's less collagen and less elastin. All right, that's it for chapter 5.